Okay, perfect.
Okay.
All right. I'd like to thank the organization committee for putting this conference together and the invitation. I am honored to be here. Um, as mentioned, my name's Anh Le-McClain. I'm an organic chemist, but I'm working as an application scientist at Magritek. And today's presentation is titled Reaction Monitoring with Benchtop NMR, Shedding New Light on Chemical Reaction Profile. So this is the overview of the presentation today. I will spend a couple minutes in the beginning to talk about why and how we can do reaction monitoring with NMR and benchtop NMR. And then the majority of the talk will be focused on different examples to demonstrate the application of reaction monitoring with benchtop NMR and what information about the chemical reaction profile we can obtain from this study. This includes observation of real-time reaction progress, the calculation of reaction kinetic data, elucidation of reaction mechanism, and optimization of reaction condition. So these examples come from Magritek's labs in both the United States and Germany, and also from our customer research program. Okay, so reaction monitor with NMR, why do we want to do it? Because NMR is a very powerful spectroscopic technique, which is a simple 1D experiment. It can provide information about chemical structure of the compounds. NMR is also inherently quantitative, which means one can do quantitative measurement on a sample without a calibration step. Lastly, NMR is considered non-destructive techniques, as sample can easily be recovered after analysis. So in case of reaction monitoring, sample can simply be returned to the reaction mixture. Reaction monitoring with benchtop or with NMR can provide real-time information without the need to work up or purify the reaction mixture. So information such as reaction progress, kinetic and conversion rate can be easily obtained. And since NMR provide information about the structural composition of the compounds, we can find intermediate, desired, and desired products. And all of this information can be used to calculate the yield and product distribution, again, without a need to work up the reaction. So on the screen here is an example of a reaction monitoring that I've done here in the lab. This is a diamine formation reaction. The stack plot down here is a 1D proton spectra collected during the reaction progress. And then integral value of plot here in the integral versus time plot. So we can observe the change in concentration of the different chemical species during the reaction. So even though monitoring reaction with NMR give a lot of useful information, it is not very easily done with the high field NMR spectrometer. So the picture here is a picture of the NMR reaction monitoring facility at the University of Bath. So the high field NMR spectrometer normally are set up in a centralized location shared by different groups. So it's not just for one lab to use. And as you can see in this picture here, the NMR spectrometer take up most of the room. There's a fume hood to do reaction. And if you look closer, there are tubings and wire to connect the NMR spectrometer to the reaction. So it is quite cumbersome to set up reaction for monitoring with a high field NMR system. So that's where the benchtop NMR system come into play. As you can see in this picture here, this is a benchtop NMR, and it fit easily in a regular size fume hood. So it can just be set up on the lab bench or in the fume hood in your lab. And you can do the reaction monitoring right there in the fume hood, since there's still plenty of space in the fume hood to set up the reactor. So it's a lot more accessible. So this is a typical setup to monitor the reaction using the benchtop NMR. On the left-hand side, that's a real-life picture of the system inside the fume hood next to the reaction flask. And on the right-hand side, this is a schematic of the setup. So we have the reactor or reaction flask, and the reaction mixture is transferred into the spectrometer using PTFE tubing. So inside the spectrometer, there's a flow cells where the sample can be analyzed, and the sample is pumped in the closed loop using a peristaltic pump. So again, the benchtop NMR spectrometer fit easily inside the fume hood, and one big advantage of doing reaction monitoring with benchtop NMR is no need for deuterated solvent, since most of the benchtop NMR spectrometer is set up with a built-in external lock, so they don't need the deuterium in the deuterated solvent to lock, which means we can do reaction monitoring using just a regular protonated solvent that you normally run this reaction with, such as ethanol or ethyl acetate. So there's no need to use deuterated solvent. So reaction monitoring with benchtop NMR can be incorporated into many different applications. We can observe the reaction progress in real time, extract and calculate reaction kinetics, perform structural analysis on different chemical compounds in the reaction mixture. And all of this information can be incorporated into slightly more complicated applications, such as online quality monitoring, reaction condition optimization, or serving as a sensor in the self-optimizing organic synthetic robots. So on this slide here is a collection of different pictures just to show how in real life the benchtop NMR system can be incorporated into different setup. So here we have the benchtop NMR setup in line with the Corning UV flow reactor to monitor or to observe the reaction progress in real time. Here's a picture of a self-optimizing organic synthesis robot. We can see many vials, liquid handling robot up here, the mass spectrometer as a sensor, and then also the benchtop NMR sitting underneath the fume hood here. Then here we have the neutralization of Muss's gas simulin. So this is another setup with a flow device. And as you can see, the balloon here is a gas using as a reactant for the reaction. Down here is a picture of the benchtop NMR set up in hyphenation with a size exclusion chromatography for real-time monitoring of the compounds coming off the size exclusion column. And then here is a picture of a process control of an industrial lithiation reaction. So box C here, this is an explosive proof box, and this benchtop NMR spectrometer sitting inside there. So it can be incorporated into a big industrial process. So with that, we will get started with different example. This first one is a simple organic reaction to learn how benchtop NMR can be used to extract reaction kinetic data. So this example came from our lab in Germany with the use of the spin salt 43 megahertz with variable temperature capability. So this is a esterification reaction between trifluoroacetic acid and ethanol. And here in the bottom, we have the stack plot of 1D proton showing the reaction mixture at the start of the reaction. We can see the quartet belong to the alcohol or the ethanol reactant. And then at the bottom is a 1D proton spectra of the reaction mixture at the end of the reaction, where we observe the disappearance of the ethanol or the alcohol signal and the appearance of the ester signal for the products. So okay, so using the spin salt 43 with the ability to analyze sample at variable temperature, the esterification was set up and monitored at different temperatures. Since an NMR is inherently quantitative, we can obtain the concentration of both ethanol reactant, highlighted in yellow trace in the plot here, and the ester product highlighted in the red trace. So the initial concentration of ethanol is about 1 molar, and we can see that its concentration decreases as the reaction proceeds, while the concentration of the ester products increases. So as the temperature increased from 28 Celsius to 60 Celsius, you can see that the rate of the reaction also increased. We can see the point of the where the concentration of product and reactants equal on this black vertical line here. So as the temperature increases, we can see that point appear sooner in the reaction progress, again signaling that the reaction rate is increasing. So with the information obtained from monitoring reaction with NMR, the rate constant of the reaction can be calculated for different temperatures shown here. So since the trifluoroacetic acid was used in Access, the reaction follows pseudo first order kinetics, which is demonstrated by the linear correlation of the logarithm of the ethanol concentration over time. Then the natural log of the rate constant are plotted with the inverse of the reaction temperature in the Arrhenius plot to calculate the activation energy of the reaction. So the ability to monitor the reaction at different temperatures allow for the calculation of these kinetic parameters. Moving on to this next example, it demonstrates the integration of benchtop NMR into the flow reactors. In this picture, we can see the benchtop NMR was set up in line with the Corning UV flow reactor. And the reaction here is a UV-induced 1,4-cycle addition shown here. This reaction is pretty typical to show the use of the UV flow reactor. The solvent in this reaction is protonated ethanol, and the concentration is pretty low, and typical for this type of the reaction is at about 100 millimolar. So here on the bottom of the page, we have the stack plot of the 1D proton spectra of the reaction mixture during the reaction progress, with the region of interest for reactant and product highlight. On the top, we have the plot of integral values of both reactant and products. And we can see that when the UV light is turned on, since this is a UV-induced reaction, when the UV light turned on, the concentration of the reactant drops significantly and the concentration of the product increase. And using the benchtop NMR to monitor this reaction, we can also observe in real time the effect of the light intensity on the reaction or on the concentration of the different species. As we can see here, different light intensity result in different concentration of both reactants and products. So this information can be used to further optimize the reaction condition. This next example is from our lab in the United States. So besides kinetic information, reaction monitoring with benchtop NMR can also help the reaction mechanism elucidation. So this is demonstrated through this cross-Aldol reaction in the synthesis of the Dibenzalacetone analog in the scheme shown here. So in this reaction, 2 molecules of 4-fluorobenzaldehyde was reacted with 1 molecule of acetone in the solvent protonated ethanol with sodium hydroxide as a catalyst. So acetone here is in the limiting reagent with 1 equivalent, whereas 4-fluorobenzaldehyde is in slight excess. So by monitoring the concentration of acetone in this reaction mixture, we can also determine the endpoint of the reaction. So here we have the proton spectra of each reactant in protonated ethanol. Solvent suppression was used to collect both of these spectra so that we can see the signal for ethanol is pretty low in intensity and comparable with the signal from the reactants. So here we have the 4-fluorobenzaldehyde in ethanol with the diagnostic aldehyde signal at about 9.5 ppm, well separated from all other signal so that we can monitor this signal during the reaction. In this green trace is acetone in ethanol, and the singlet here at around 1.5 ppm correspond to the acetone, and as we can see, it is overlapping with some other signal. These are the C13 satellite coming from the ethanol solvent. So with that, we decided to monitor the reaction using both solvent suppression and C13 decouple. And with that, we can see the acetone signal is nice and clear to be integrated for monitoring. And since acetone is a limiting reagent, we need to know when acetone disappears from the reaction mixture for the endpoint of the reaction. So this is a stack plot of the 1D proton spectra of reaction mixture as reaction proceeded. The acetone signal or singlet is highlighted in red with the red trace over here in the integral versus time plot. So we can see that the signal decreases as the time goes on during the reaction, and then toward the end it approaching zero or remain undetectable in the reaction mixture. The blue signal here correspond to the vinyl proton in the final products. And in the plot here, we can see its concentration increase as the reaction goes on. So monitoring this reaction using benchtop NMR, we also observe one singlet here highlighted in green trace next to the acetone signal. Its concentration, if we look in the graph here, increase at the beginning of the reaction and then decrease toward the end of the reaction. So this suggests that the green signal belong to an intermediate species as they get produced at the beginning of the reaction and then get consumed as the reaction continues. So this is the intermediate species that we observe in monitoring of this reaction. So in conclusion for this example, monitoring reaction with benchtop NMR allow for the observation of the intermediate species, which sometimes can be difficult to isolate and analyze. So this helps to determine the mechanism of the reaction of interest. So beside kinetic data and reaction mechanism, using benchtop NMR to monitor the reaction also allow for the analysis of the reaction product mixture. So in this example, this came from our lab in Germany. The stereoisomer ratio of the product mixture in the Horner-Wadsworth-Emmons reaction here can be determined directly in line without a need for separation or calibration curve. So these two products are stereoisomer, and sometimes they can be fairly difficult to separate. So this is a HW reaction with diethylphosphoacetic acid and 4-nitrobenzaldehyde. The product of the reaction are both E and Z acrylic acid as shown here. So the HWE reaction can be stereoselective to form mainly the E products when the T-meta chemical is used or is added to the reaction mixture. So using NMR to monitor this reaction, we can not only observe the reaction progress and obtain the kinetic data, but we can also determine the ratio or the amount of each product's form. So these stack plots show the 1D proton spectra of each reactant and the reaction mixture. The solvent used in this reaction was protonated THF, and you can see the THF highlight here in blue boxes. And we observed this reaction with the solvent suppression on. So the intensity of the THF signal here are very comparable to the intensity from the analytes. This diagnostic signal for aldehyde was used to measure to follow the reaction progress. And then also the signal highlight in gray here, this is the region of interest to analyze both of the E and Z products. All right, so this is a 1D proton spectrum of the reaction mixture without T-meta. So without T-meta, we would observe the formation of both E and Z products. We can identify signal belong to the vinyls group of the E products here with a coupling constant of about 15.9 hertz. And then the Z products can also be identified with a coupling constant of 12.7 hertz. And we can quantify the concentration of each of them that come out to be 79% of E and 21% of Z. And this is comparable to the literature value reported. So this is a 1D proton spectrum of the reaction mixture with T-meta. So with T-meta, we can observe mainly the E isomer as a major product, and the ratio come out to be greater than 95 of the E to 5% of the Z products, which is also comparable to the literature reported value. So this example was monitored on our 80 megahertz ultra, and it demonstrates the use of the benchtop NMR system to not only follow the course of the reaction during the online monitoring reaction experiment, but also just to study the chemical or the stereochemical outcome of this type of reaction. Since NMR can give both structural and quantitative information, we were able to determine both isomer products of the reaction and quantify the ratio, again without the need of working up the reaction mixture or separating the two isomers. So this result also agrees very well with the reported values in the literature where they actually had to separate the two isomers. All right, so the next example here using benchtop NMR to optimize reaction conditions come from one of our customer, the group in Jülich, Germany. So they developed a biocatalytic approach to synthesize aromatic amino alcohol stereoselectively in a cascade reaction with two consecutive steps. The first step used the enzyme called carboligates to convert the hydroxybenzaldehyde to hydroxy phenylpropanone here with the co-substrate of pyruvate and the side product of CO2. The second step used a transaminase enzyme with an amine donor to install the amine moiety in the final products of hydroxypropylphenol. So their aim in this project was to find the optimal balancing amount of substrate and catalyst for this reaction. Since enzymes are quite picky in terms of reaction condition and concentration, so it's very crucial to find the optimal condition and amount of catalyst. So the main goal here was to determine the optimal time point where the first step of the reaction is complete to start the second step. So as you can think, if the first step is not quite finished and we start the second step too soon, we can observe substrate inhibition of the enzyme or side product getting produced. So these are the challenges that they were facing, and they use a benchtop NMR system in this pretty typical setup for online reaction monitoring for the kinetic study of this cascade reaction. So here's a kinetic study that can be separated into two steps. The first step is shown here for the carboligation reaction, which took about 2 hours, as the plot shown here at 30 Celsius. So in this stack plot, we can see many different signals, but what stand out the most is this broad signal here at about 4.7 ppm. So this belongs to the water in the buffer that was used in the reaction. But in this example, they did not need to use a solvent suppression since all of the region of interest is well separated from this buffer or the water signal. So in this plot here, we can see the signal box in black. This is the diagnostic signal or the proton signal in the aldehyde starting material. And as we can see, as time goes on, its concentration decrease. and it is plotted over here in the concentration in millimolar over 10 minutes at the black trace. So starting out, we can see the concentration was at about 50 millimolar. At about 30 minutes of the reaction, the concentration dropped down to about 5 millimolar before it remained undetected. So the benchtop NMR in this instant can detect in the single-digit millimolar concentration. Then we have the signal highlighted in purple or violet here. So this signal belonged to the pyruvate co-substrate, and it was used in access, as we can see in the plot here, that it leveled out about 60 millimolar. And then the last signal is a singlet at around 2 ppm, box in gray. This is the methyl group in the product for this step, and its concentration is plotted here, and we can see the concentration increase as the time increases. So for this compound, they also do offline HPLC analysis as highlighted in orange dot, and we can see the gray and orange correspond very well together, which means that the concentration monitored by the benchtop NMR agree very well with the HPLC measurement system. One thing I forgot to mention was that they also use a internal calibrant. The signal appear at 0 here, and this is what allowed them to do the absolute quantification to report these concentration values over here. All right, so this is the plot for the second step of the reaction, the reductive amination step. Again, we can see the big water signal coming from the buffer, but the region of interest is well separated from this buffer signal. So here in the gray box, this is the signal from the methyl group, the product from the first step, and now it is the reactant in the second step. And we can see its concentration decrease as time goes on. We can also observe its trend line over here in the concentration versus time plot as it's increased. This reaction took a lot longer at around 24 hours. The signal box in green or highlighted in green, this is from the amine transfer as a co-substrate in access. So we can see its concentration decrease, but it did not go to 0 since it was in access. And then the signal here, there are 1 ppm box in black. This is the signal corresponds to the methyl group at the final products. And we can see its concentration increase to about 20 millimolar at the end of the reaction. Again, they also took offline HPLC measurement highlighted in orange signal, and the concentration value both measure in NMR and HPLC agree very well with each other. So overall, in this example, benchtop NMR enable the researcher to detect concentration of different components in a buffer solution down to single-digit millimolar concentration range without a solvent suppression technique. The concentration obtained from the benchtop NMR were in well agreement with the offline HPLC measurement. And so they could achieve the goal to determine the optimal time point to start the second step of the cascade reaction after around 45 minutes of the first step, so that they can achieve the highest overall yield for the cascade reaction. So their goal for the future would be to set up a self-regulation system with a feedback loop to optimize their biocatalytic cascade reaction. All right, so this coming up example showcase the use of benchtop NMR as a compact and mobile inline sensor for oxidative neutralization of Mercer's gas simulant from a group at the University of Nantes in France. So their goal was to develop a simple and portable continuous flow device shown here in the picture to effectively neutralize Mercer's gas chemical warfare using an inline benchtop NMR to monitor and adjust the reaction condition in real time. This small flow reactor device, along with the compact mobile inline NMR sensor, can be developed by the police or the military force, and it can be deployed to the field and perform the neutralization directly on site. So here's the chemistry behind the setup of this device. Mustard gas, or HD, in this scheme can be neutralized by oxidation to a less toxic sulfoxide species. This oxidation needs to be selective, since overoxidation results in a highly toxic sulfone species. So the process of neutralization of mustard gas is generally conducted under a harsh conditions, such as direct pyrolysis or hydrolysis with strongly basic solution, or transformation with very aggressive oxidants. So this group developed a oxidant procedure using pretty mild oxidant to neutralize the mustard gas simulant. So this is just very similar to mustard gas, we just have the molecule. So they are CEPS or CEES. Again, they use very mild oxidant and can be set up in a portable and flow device as shown here. So the fully selective sulfoxidation of mustard gas simulant, CEES or CEPS, can be carried out with a mixture of oxidants consists of urea and hydrogen peroxide adducts, and methanesulfonic acid and methanol. They employ a Taylor NMR pulse sequence, the wet 180 nosy pulse sequence, to suppress the multiple solvent resonance at low magnetic field under the flow condition. So this stack plot here shows two 1D proton spectra recorded at different resident temp at 33.6 second and 67.2 second. So the signal for this sulfoxide product CESO can be observed here at around 3.8 ppm. And it's nice and separated, easy to integrate. At 1.25 ppm here, we can see an overlapping signal of CEES. This is the mustard gas simulant, and CEESO, this is the sulfoxide product. So they overlap at the beginning or at around 33 seconds of resident, but at 67 seconds, we can only observe one set of triplet, which means the CES has been completely used up. So as the conversion rate as a function of time can be plotted over here, and as we can see at around 70 seconds, the CES has been completely oxidized to the CEESO, which is pretty phenomenal, since the full conversion can be reached, which is then less or just a little bit more than a minute, stressing the efficiency of the neutralization method. So with the use of the compact and mobile benchtop NMR spectrometer, the method for complete and selective sulfoxidation of the mustard gas simulant was developed and was shown to be fairly efficient and practical, and now they can be boxed up and deployed for the field intervention. So this is the last example that I'd like to discuss, and this is the use of benchtop NMR system as an online sensor for organic synthesis robot. So Dr. Lee Groning's lab at the University of Glasgow in Scotland has pioneered in the field of the robot-driven detection of novel organic chemical reaction. So artificial intelligence work in this closed loop system to explore the chemical space. A liquid handling robot performed the experiment or the reaction, so mixing different reagent and solvent. Then the reaction mixture is analyzed using online analytical technology, both mass spec and NMR spectrometry. This data can then be processed this data can then be processed using the artificial intelligence to extract information about the reactivity, and it is used to create a model or the chemical space to suggest the next experiment parameter for the robot to perform. So the one outcome of this organic synthesis robot was a multi-step single substrate cascade reaction shown here. So this is a new mode of reactivity for the methylene isocyanide. So in this reaction, p-toluene sulfomethyl isocyanide in presence of an activator, in this case is diethyl bromomalonate, reacts consuming 6 equivalents of itself to yield a trimeric product with in high yield of 47%. This is without optimization. This product or this reaction show a formation of 5 new carbon-carbon bonds. So a chemical informatic analysis revealed that this transformation is both highly unpredictable and able to generate an increased complexity like a one-part multi-component reaction. So hopefully I have shown you an example that demonstrate all the difference and diverse application using Benchtop NMR to monitor the reactions. With the compact and mobilized mobile Benchtop NMR system, we can obtain different information about the reaction ranging from kinetic data to structural analysis to stereochemical outcome. Furthermore, the Benchtop NMR system can also serve as an inline sensor for the different flow device and self-optimizing robot. And with that, I'd like to say thank you to our partner and customer of whom I've shared the data today. And thank you for your attention. I'll be happy to answer any questions.